Hello, hello. In this chapter, we will elaborate on the metabolism of the nutrients that we met in chapter 2 and look at the concept of nutrition. This is going to be very superficial. Most of it should be review, especially if you took a very good biology or biochemistry course elsewhere. So you should see some familiar terms, and don't worry, it won't be too heavy. Oh, for the eighth time, what is metabolism? All the chemical reactions taking place in the body. So we talked about catabolic reactions, which break down large molecules into smaller ones, versus those anabolic reactions that build up larger molecules by combining smaller ones. Now, metabolism is a balance of these reactions. ATP is the intermediate molecule that captures and releases energy during these reactions. So ADP, which has two phosphate groups, can be attached to an inorganic phosphate or a P when there's available energy captured during enzymatic reactions, and that forms ATP. ATP can then be broken down to release that energy to be used for various processes, and then that results in ADP and inorganic phosphate. So these reactions are reversible, and ATP is the energy currency of your cells. We mentioned the interplay between ATP and ADP and phosphate as intermediate molecules in metabolic reactions, anabolic and catabolic. Let's begin with the purple box at the top. Simple molecules such as glucose, which is the basic unit of your carbohydrates, or amino acids, which are the building blocks of your proteins, or glycerol and fatty acids, which are the components of triglycerides, can be linked together through covalent bonds during anabolic reactions. This requires a transfer of energy from ATP to form the bonds within these molecules. This results in complex macromolecules such as glycogen from glucose, proteins from the amino acids, and triglycerides from the glycerol and fatty acids, which essentially are the storage forms and the functional forms of these molecules. When energy is required from these molecules, or the building blocks are required for other processes, these large molecules can be broken down in catabolic reactions, where the energy in the bonds will then be transferred to ATP by linking ADP and phosphate. And that breaks them down into their smaller molecules. So there's this constant interplay between the smaller molecules and the larger storage forms, as well as anabolic and catabolic reactions. Note that heat is released during all of these processes. So whenever we talk about metabolism, we also have to consider heat production. Let's consider the metabolism of carbohydrates. Since carbohydrates, as you recall, are the preferred energy source for most cells of the body, carbohydrate metabolism really is about metabolism of glucose. As we saw, all of the carbohydrates, including the polysaccharides and disaccharides, have glucose, and the other monosaccharides, fructose and galactose, can be converted into substrates that enter the glucose metabolism pathway. So again, metabolism of carbohydrates is really about metabolizing glucose. The body's use of glucose depends on the needs of the cells at the time. So the first need that is met is ATP production. Glucose will be converted into ATP to meet the energy demands of the cells. Then certain amino acids can be synthesized from glucose. Excess glucose is stored as glycogen by linking multiple glucose units together. Glycogen is stored in the liver as well as your muscle tissue. And then excess carbohydrates, excess glucose, can be converted into triglycerides, and our bodies again can store triglycerides without limit. So be careful. Carbs are necessary, but you don't want them in excess. In order for cells to use glucose, glucose must pass out of the blood into the interstitial fluid around the cells and then be taken up into the cells by crossing the plasma membrane. Glucose is polar and relatively large, so it requires a little help to enter these cells. This is performed by a process called facilitated diffusion, where protein transporters in the membranes of these cells move glucose into the cell. These glucose transporters, or GLUT proteins, are present in the membrane of almost every cell in the body. The hormone insulin increases the number of GLUT4 transporters in the membrane of muscle and adipose cells, increasing the uptake of glucose out of the blood into these cells. Note that exercise also increases the number of GLUT4 transporters in these cells, thereby promoting glucose uptake. So both insulin and exercise help to lower blood sugar levels. So the oxidation or the breakdown of glucose to produce ATP occurs in processes collectively called cellular respiration. So this involves 4 sets of reactions in your cells, beginning with glycolysis, followed by formation of acetocoenzyme A. Acetocoenzyme A then enters a series of reactions collectively called Krebs cycle. And finally, the products from glycolysis, formation of acetocoenzyme A, and Krebs cycle enter the electron transport chain where most of the ATP is produced. So we're going to look at each of these steps. We're going to see where they occur in the cell. We are also going to look at what products come out of each set of reactions and the overall effect with respect to ATP production. Here we have an overview of glucose metabolism. Once glucose enters the cells, it is quickly phosphorylated and enters glycolysis. During glycolysis, which occurs in the cytoplasm of the cell, the 6-carbon glucose molecule is converted into 2 3-carbon molecules called pyruvic acid. During these steps, small amounts of ATP are produced, net amount of 2, as well as reduced coenzymes NADH. So the energy from the breakdown of glucose is temporarily stored in the reduced coenzyme. If there is enough oxygen in the cell, pyruvic acid enters the mitochondria and is converted into acetocoenzyme A, which has 2 carbons. This means that a carbon was removed in the form of carbon dioxide. This process also produces some more reduced coenzymes. Acetocoenzyme A then enters a series of reactions called Krebs cycle, which produces a little more ATP, more carbon dioxide, and several reduced coenzymes. The carbon dioxide is a waste product produced during cellular respiration. That's why we exhale carbon dioxide as a waste. The reduced coenzymes from the first 3 steps then enter the electron transport chain, where several molecules of ATP are produced. Water is a byproduct, and this is where oxygen plays a direct role in carbohydrate metabolism. But note that the pyruvic acid would not even enter the mitochondria to begin formation of coenzyme A or Krebs cycle if oxygen is insufficient. Again, glycolysis is a process where the 6-carbon glucose is split into 2 3-carbon molecules of pyruvic acid, generating a small amount of ATP, a net of 2, as well as reduced coenzymes. All right, summarized here. We skipped all the little steps, so again, you do not need to know the names of all the intermediate molecules. You should be happy about that. The fate of the pyruvic acid following glycolysis depends on the availability of oxygen. If there is insufficient oxygen in the cells, those conditions are called anaerobic, without oxygen. And here, pyruvic acid is reduced into a waste product called lactic acid. And the buildup of lactic acid acidifies the cells and interferes with energy metabolism. So that might manifest as muscle cramps or decreased endurance. However, if oxygen is plentiful, and the cells are under aerobic conditions, most of the pyruvic acid will enter the mitochondria and be converted into acetyl coenzyme A. So notice that glycolysis does not require oxygen, so it is produced in the absence of oxygen or in the presence of oxygen. But Krebs cycle and the electron transport chain following the formation of acetyl coenzyme A can only occur under aerobic conditions. Here we have the pyruvic acid produced from glycolysis entering the mitochondria to be converted into acetyl coenzyme A only under aerobic conditions. If there is inadequate oxygen and the cells are under anaerobic conditions, the pyruvic acid will be reduced to lactic acid. When pyruvic acid is converted into acetyl coenzyme A, carbon dioxide is produced as a waste, as well as some more NADH, a reduced coenzyme that temporarily stores the energy from those reactions. Acetyl coenzyme A then enters Krebs cycle, where more carbon dioxide is produced, as well as several reduced coenzymes and small amounts of ATP. Krebs cycle was formerly known as the citric acid cycle. It occurs within the matrix or the inner part of the mitochondria. It is a series of reactions that begin with the 2-carbon substrate acetyl coenzyme A. And also note, please, that acetyl coenzyme A is an important intermediate in several metabolic pathways, including fat metabolism as well as protein metabolism. So as we summarized, carbon dioxide is produced as well as reduced coenzymes, NADH and FADH2, and 1 molecule of ATP per pyruvate, but remember you get 2 pyruvates per glucose molecule, so a net amount of 2 ATP are produced during Krebs cycle, which is depicted here. And the reduced coenzymes are going to enter the electron transport chain, while carbon dioxide is going to be released as a waste during respiration. The electron transport chain occurs in the membrane of the mitochondria. This is where oxygen acts as the final electron acceptor, producing water as a byproduct, and the major outcome is the production of several molecules of ATP. These reactions involve the movement of hydrogen from molecule to molecule. Don't get into the details too much, just work with the summary. So there's an overview of the carbohydrate metabolism. Please note the 4 steps and what is produced during each step. Once the cellular's ATP needs are met through cellular respiration, excess glucose will then be converted into glycogen and stored in the liver and muscle, as we've mentioned before. This process is stimulated by insulin, and the process is called glycogenesis. Genesis meaning synthesis or formation. So glucose is converted into glycogen in the process called glycogenesis. This occurs when glucose levels are high in the blood, usually right after a meal. That is called the absorptive state. So once you've absorbed glucose from your food, blood sugar levels tend to rise. This stimulates the release of insulin, which promotes the uptake of glucose out of the cells, but also stimulates the pathways that convert glucose into glycogen. That is glycogenesis. A few hours later, when blood glucose levels are low because the cells have taken up and used glucose, homeostasis will stimulate the release of glucose from the storage forms into the blood. The breakdown of glycogen to release glucose is called glycogenolysis, lysis, the breakdown of. So this is a catabolic reaction. Glycogenesis is anabolic. glycogenolysis is catabolic. This process is inhibited by insulin and promoted by insulin's counter-regulatory hormones, including glucagon, epinephrine, norepinephrine, as well as cortisol. This occurs during the post-absorptive state, or in between meals, when blood sugar and other fuel levels tend to decline. During the post-absorptive state, when glycogenolysis is stimulated, more glucose can be produced once the glycogen stores have been depleted. This involves the formation of new glucose molecules from non-carbohydrate substrates. The process is called gluconeogenesis, or the formation of new glucose. It is performed by the liver, and the substrates that can be converted into new glucose include lactic acid, certain amino acids, and glycerol from triglycerides. So fats can be converted into glucose. Look at that. As well as lactic acid and certain amino acids. This is stimulated again by cortisol and glucagon, which are active during the post-absorptive state when blood sugar levels tend to be low. So these hormones work to raise blood sugar levels back into normal homeostatic range. And these hormones and this process counteracts the effects of insulin. So insulin is active when blood sugar levels are high and need to be brought down. Insulin promotes the uptake of glucose out of the cells and conversion to their storage forms, while cortisol and glucagon counteract insulin by promoting the breakdown of these stored nutrients and the release of glucose into the blood. We will now move on to lipid metabolism. Recall that lipids are hydrocarbons that are nonpolar and therefore are hydrophobic. They do not dissolve well in water, but water is the main component of blood. So the lipids have to be made transportable somehow. Lipids are made soluble by combining with polar proteins. So amino acids are very polar. They have a carboxyl group. They have nitrogen, which makes them very polar. So by combining proteins with lipids, the lipids now become transportable in the blood and other fluids of the body. The combination of these proteins with lipids creates transport molecules called lipoproteins. Here's a picture of a lipoprotein. What you're seeing in blue and green are the phospholipids, and they are surrounding a core of triglycerides. Remember that phospholipids have that polar head created by the phosphate group, so they help to also make lipids transportable. But the core of the hydrophobic triglycerides, as well as fat-soluble vitamins, are surrounded by phospholipids as well as these apoproteins that help to make the lipids soluble or transportable in the blood plasma. Depending on the type of lipoprotein, they will have different percentages of the various fats, including triglycerides, cholesterol, phospholipids, and vitamins. There are four major classes of lipoproteins that transport lipids throughout the body in the blood. Number one are the chylomicrons. These originate in the digestive tract and are packages of dietary lipids that have been absorbed into the intestine. They are then combined with proteins and eventually enter the bloodstream, where they travel throughout the body, releasing triglycerides to adipose tissue for storage. Eventually, chylomicron remnants are taken up by the liver, and the components of the chylomicrons are repackaged. The liver then can synthesize another lipoprotein called very low density lipoproteins, which tend to be high in triglycerides but also contain cholesterol. As very low density lipoproteins travel in the blood throughout the body, they also release triglycerides for storage in adipose tissue. As they release the triglycerides, their density changes and they become intermediate density lipoproteins and eventually low density lipoproteins or LDLs. Now, because these have released triglycerides, they are now high in their concentration of cholesterol. And LDLs travel in the blood and release cholesterol to the various cells of the body. So we saw the importance of cholesterol for hormone synthesis and the synthesis of other molecules, as well as plasma membrane formation. However, when LDL, or this form of cholesterol, is in excess, it can lead to the development of atherosclerosis, where the fat builds up in the artery walls and limits blood flow to the tissues. That leads to cardiovascular disease, including heart attacks, stroke in the brain, and peripheral vascular diseases. So LDL has gotten a bad rap over the years and is designated bad cholesterol. So you want to make sure your LDL levels are low by adjusting the diets and minimizing saturated and trans fats. High-density lipoprotein is also produced by the liver, but its role is opposite to that of the low-density lipoprotein. HDL actually travels in the blood and collects excess cholesterol, returning it to the liver for elimination in the digestive tract. So HDL has been dubbed good cholesterol because it removes excess fat, reduces the risk of atherosclerosis and cardiovascular disease, and HDH levels tend to increase with a diet that is higher in polyunsaturated fats versus the trans and saturated fats. Genetics also play a role in one's lipid profile, and individuals can have great dietary practices and even exercise and reduce their fat levels, but still have a poor lipoprotein profile due to genetics. So for those individuals, medications may be necessary to adjust their blood fat levels. Cholesterol, we acquire it from some foods such as eggs, dairy, and meats. But note that most of the cholesterol is synthesized by your liver cells or hepatocytes. So we just saw that an increase in total cholesterol levels are associated with a greater risk of coronary artery disease. And as we mentioned, exercise, diet, and certain drugs can be used to reduce cholesterol levels. In terms of lipid metabolism, lipids can be oxidized to produce ATP. So the main purpose of your triglycerides is to release the fatty acids that can be used to make ATP. We saw that excess carbohydrates and excess lipids themselves can be stored in adipose tissue without limit. So while lipids are necessary in your diet, they should be limited, and your best form of lipids or triglycerides are your polyunsaturated fatty acids, including those essential fatty acids. Lipids also can be used for structural molecules, and some can be used to synthesize other essential substances. This table we saw in the previous chapter. It is a summary of the types of lipids in the body and their functions. Just a review for you there. Triglycerides constitute 98% of one's energy reserves and therefore is how the body stores most of its energy. Triglycerides are acquired from chylomicrons and VLDLs and are stored in adipose tissue. Now, when blood fuel levels are low, as in the post-absorptive state, triglycerides are broken down or catabolized in a process called lipolysis. This releases fatty acids that can be oxidized for ATP synthesis, as well as glycerol for gluconeogenesis, making new glucose. Lipolysis is stimulated by the hormones epinephrine and cortisol, but is inhibited by insulin. Insulin, however, stimulates lipogenesis during the absorptive state, or lipid anabolism, which is a process of synthesizing triglycerides from fatty acids and glycerol, or glucose, or amino acids. Lipogenesis occurs whenever blood lipid or other nutrient levels are high. Here we have an overview of lipid metabolism. Please note the triglycerides on the right of the slide can be broken down into glycerol and fatty acids in the process called lipolysis, which again is stimulated by epinephrine, norepinephrine, cortisol. That will provide substrates for gluconeogenesis as well as beta oxidation, where acetyl coenzyme is formed, which then leads to the formation of several ATP molecules. The opposite reaction is lipogenesis, which is stimulated by insulin, where glycerol and fatty acids are converted into the storage form of the fats, the triglycerides. Also note that under conditions of extreme nutrient deprivation or starvation, acetocoenzyme A can also be converted into products called ketone bodies in a process called ketogenesis, which occurs in the liver. And these ketone bodies can be used as nutrient substrates for ATP production in certain cells of the body, particularly the brain when glucose is insufficient, as well as red blood cells, if I'm not mistaken. Also note that certain amino acids can be converted into acetocoenzyme A for energy production, or again, through lipogenesis, can be converted into triglycerides when in excess. Let's move on to proteins. When proteins are digested, they are broken down into their units called amino acids, which are absorbed. Amino acids can be oxidized to produce ATP or can be synthesized to make new proteins, which is the main purpose of amino acids in your diet. Okay, so remember that amino acids are only used for energy under extreme conditions. Many proteins function as enzymes, which are biological catalysts, which essentially allow the reactions of the body to occur. But we saw the other functions of proteins, which are summarized on the next slide. So proteins can be structural, regulatory, contractile, provide protection through immunity. They can also transport, as we saw, for example, with lipoproteins, or the main function, again, act as catalysts. So different proteins have different functions, and you also recall that each protein's function is determined by its amino acid sequence, which is determined by the genes in the DNA that code for the proteins. So we mentioned before that amino acids can be oxidized to produce ATP, but mainly are used to synthesize new proteins. Excess amino acids can be converted into glucose when necessary and stored as triglycerides during lipogenesis. Now, in the breakdown of proteins during cell breakdown or where proteins are defective, before they can be used for gluconeogenesis, the amino group must be removed. That process is called deamination, and it occurs in the liver. That amino group produces a waste called ammonia, which is toxic. So the liver converts ammonia into urea, which then is excreted in the urine. So this is how we trace the nitrogen in the urine that can be used to assess nitrogen balance. Now, synthesis of proteins is carried out by the ribosomes of your cells. We acquire proteins from our diet. Of the 20 amino acids, 10 are considered essential, meaning that your body needs them to make proteins, but your body cannot synthesize these amino acids from other substrates, and therefore they must be acquired in the diet. The other amino acids are non-essential and can be synthesized by a process called transamination. Now, regarding these essential amino acids, you got to get them in your food. Certain food types provide all 10 essential amino acids and therefore are called complete proteins. Again, they provide sufficient amounts of all the essential amino acids, and these are mainly animal proteins. So proteins found in beef, fish, poultry, eggs, etc. Meat proteins tend to be complete proteins because they provide all the essential amino acids. Now, plant proteins tend to be incomplete, meaning they are deficient in one or two of the essential amino acids. This becomes an issue for those that do not eat any meat products or the absolute vegan. So how do they acquire all of their essential amino acids? Interestingly, certain types of plant proteins contain most of the essential amino acids but are lacking one or two, and another group of plant proteins contain the missing essential amino acids that the other group doesn't have. So if those groups of plant proteins are combined in the diet, they then will create a full complement of the essential amino acids. So plant proteins tend to be incomplete because they do not contain sufficient amounts of all the essential amino acids. But when plant proteins of different groups are combined, they then provide all the essential amino acids and now are called complementary proteins. So these groups include the leafy green vegetables, legumes, and grains. By combining legumes in particular, such as peanuts, beans, lentils, chickpeas, peanuts, those are legumes, with grain proteins such as rice and corn and wheat and barley, that will provide the full complement of essential amino acids. So nutrients in general are chemicals in food that our bodies use for growth, maintenance, and repair. We looked at the organic molecules, the carbs, lipids, and proteins. Nutrients also include water, minerals, and vitamins. You need a lot of water, but only trace amounts of minerals and vitamins. So it is recommended that your calorie distribution look like this. 50 to 60% of your calories or the source of your energy in your food should come from carbohydrates. But most of those carbohydrates should be the complex carbohydrates or your polysaccharides, including lots of fiber. That will minimize the release of insulin and the anabolic effects that lead to weight gain. Okay, so minimize your simple sugars. 30% of your calories should come from fats, or less than 30%. And you want to minimize the saturated and trans fats, which increase the risk of cardiovascular disease. And that leaves about 12 to 15% from protein. So the U.S. Department of Agriculture introduced a concept called My Plate, which we will see on the next slide, to emphasize what your food portions should look like relative to each nutrient class. So each type of food provides different amounts of each nutrient. For example, fruits are high in sucrose, a simple sugar. They may have some starch, but fruits also contain a lot of water, vitamins, and fiber. So fruits should represent a relatively significant amount of your diet. The highest proportion should come from vegetables. Vegetables provide starch as a complex carbohydrate, lots of fiber, and lots of vitamins and some minerals. Okay? Protein foods include meats from animal muscle. Protein foods not only provide protein, but they come with some fats as well as some vitamins, especially your fat-soluble vitamins. Your grains provide, again, complex carbohydrates, some maltose as a simple carbohydrate, and small amounts of some vitamins. Dairy provides fat in the form of saturated fats. It provides carbohydrates in the form of lactose, as well as fat-soluble vitamins. So Look at the proportions and hopefully you'll adjust your diet and read labels to ensure that you're getting the proper balance of all these nutrients. Minerals are inorganic elements that are important for reactions and only are required in small amounts. So if you eat healthy foods, natural foods, less processed foods, you'll get plenty of minerals. Vitamins are also required in small amounts and are required for metabolism. They cannot be synthesized by the body naturally, but must be consumed in food, so they are essential nutrients. I'm going to qualify that. The bacteria that colonize your large intestine can produce small amounts of two vitamins, B vitamins, B12 in particular, and vitamin K. But that's not you. That's the bacteria producing those. So for all intent and purposes, your vitamins need to come from your diet. Vitamins can be classified as fat-soluble versus water-soluble. Fat-soluble vitamins, again, are A, D, E, and K. They also must be ingested and absorbed with other fats. Water-soluble vitamins include vitamin C and all the B vitamins. Fat-soluble vitamins not only require fat for absorption, they can also be stored in the soft tissues of your body, especially the liver, if they are consumed in excess and can lead to vitamin toxicity. Water-soluble vitamins are not stored and excess amounts are flushed out in the urine. Antioxidants are vitamins A, C, and E. These vitamins work together to remove harmful free radical molecules from your body. Free radicals or reactive oxygen or nitrogen species are unstable molecules that damage cell membranes, other intracellular structures, including the DNA, and can lead to mutations implicated with aging as well as cancer. So we want to increase the amount of vitamins A, C, and E in our diet to limit the amount of free radicals. Please go through this table and quickly note the sources and functions of each of the vitamins. But the main thing to focus on is the classification as either fat-soluble, water-soluble, or antioxidant. All right? Read your labels when you eat, when you shop, and do your best to ensure you have a healthy, balanced diet.